Today we'll be talking about Norrie disease. So, what's the whole point of a signaling transduction pathway? The point of a signaling pathway is to bring about a cellular response to a signal from outside the cell. It's vital for organisms to be able to sense and respond to the environment. These molecular events within the cell help to regulate homeostasis. Homeostasis is basically when an organism is able to maintain the internal environment of the body. For example, when it's hot outside, we naturally start to produce sweat. This is how your body responds to a change in its environment. The same happens at a molecular level. It's important to realize that cells get numerous signals, but they can only respond to just a few. This is because a cell's receptor determines whether or not the cell will respond to a signal. Also, receptor proteins are very specific to its signal. The pathway involves primarily three steps: reception, transduction, and response. The first step starts off with a ligand, or a chemical messenger, which binds to the active site of a receptor protein. Because the receptors are very specific to its signal, we can think of it as a one-to-one relationship, where one receptor is associated with only one signal coming from outside the cell. When a ligand binds to the receptor, it results in a change in the receptor conformational 3D shape. These interactions are reversible, meaning that the two molecules will join together and come apart together. Reversibility is important because if the ligand was never released, the receptor would be continuously stimulated. Also, we should keep in mind that an inhibitor may also compete to bind to a receptor protein, which may alter human behavior binding and prevent the binding of receptor-specific ligands. The second phase, which is transduction, is to be able to convert the signal so the cell can respond to it appropriately. In this image, we see that the membrane receptors are protein kinase receptors that become activated through the hydrolysis of ATP. We can think about it as a reaction, and on the reactant side, there is an inactive protein in ATP. With the help of enzymes known as protein kinases, it results in ADP and active protein. In other scenarios, the protein kinase acts as a relay protein, which is activated by the binding of a secondary messenger to speed up the next phosphorylation that results in a more magnified cells. Also, secondary messengers lie in this step as well. They are not enzymes. Instead, they are simply chemical messengers that get into the cell through the means of a protein. Their job is to regulate target enzymes by binding to their active site so the signal can be distributed and amplified in the cell. This also results in a conformational change in the protein shape as well. Finally, we have our response. This may be short or long-term depending on the signal being carried out through the cell, but some of them include gene expression, the switching off of genes, apoptosis, which is programming cell death, alteration in an enzyme, and much more.
After learning about the function of the cell pathway, let's put everything into the context of a real disease. An example is Norrie's disease. This is a genetic eye disorder that occurs due to a mutation of the NDP gene in the X chromosome. It affects male infants before or after birth and causes irregular development of the retina. Symptoms include blindness, cataracts, which is clouding the lens like the image shown, irises or eyeball shrinking after birth, and more. The WNT pathway starts when the NDP gene encodes norrin. Norrin is a protein that involves in the formation of new blood vessels of the retina. This protein binds with WNT, a ligand that's important for cell growth, and activates FZD4, a receptor. LRP5 provides instructions for making proteins and helps FZD4 to split up GSK3 and axin for the B-catenin. The split up allows the B-catenin to be phosphorylated and stabilized before entering the nucleus to bind together with other factors of the mRNA. This pathway helps develop sight and hearing. However, an incorrect pathway can lead to Norrie's disease. A mutation occurs in the NDP gene and is unable to encode norrin, which leads to a defective norrin. Without the norrin, blood vessels can't be formed. The FZD4 is not activated and can't split up GSK3 and axin from the B-catenin. So the B-catenin is ubiquitinated, which means it is marked and can undergo proximal degradation, which is able to split up from the proteins. Through this process, the B-catenin is not stabilized and can't enter the nucleus. So defective norrin is the result of Norrie's disease. As of right now, there is no cure because there's no way to reverse the loss of vision and hearing loss. However, a group of researchers in the University College London is working to find a solution to prevent or slow down hearing loss. Another is using gene therapy and is testing on animals, but they are still in the early stages. Also, they are recruiting patients with Norrie's disease to be part of their medical trials to gain data and hopefully learn more about its function and make efficient treatments. Thank you for listening.